I'm Nadia Soudzilovskaia. I'm professor ecosystem functioning at Hasselt University and director of Ecotron research facility. I'm also representing here the ANAIS, the same infrastructure that Ilya was talking about. So ANAIS stands for Analysis and Experimentation in Ecosystems. And in this particular case, my team is working on understanding phenomena of soil phenology and soil phenological mismatch in soil using the Ecotron setup. So we are doing a lot of work in this direction, but today I will specifically focus on studies and data and questions which we have in relation to the PHENET project. So what is phenological mismatch? We know that climate change affects ecosystem functioning. It affects actually every organism in the ecosystem in various ways, and it also affects the phenology of the organisms, which means that in changing climate, plants are starting, for instance, to flower earlier, birds are arriving earlier or later, insects, timing of insects is changing. So all these manifestations are impacts of climate change on phenology. And what is also very important effect of climate change that it does affect phenology of individual organisms, but these impacts can be very different. So while some organisms, timing of some organisms is affected, of other organisms it may be not affected. And especially for soil food webs and ecosystem functions related to soil functioning and functioning of food webs in soil, there is very little known about the phenology in principle, and even more about phenological mismatches. So basically, we don't know if plant above ground organs are affected by climate change. For instance, returning to my example, plant starts flowering earlier than it should do. Does it affect root functions? Does it affect function of root-associated organisms? We don't know. So historically, studies on phenological mismatch has been focusing on plants, a little bit on relationships between plants and plants, insects, birds, and other animal interactions, and interactions in soil. A relationship between plants and soil is very, very poorly understood. So to address this question, we are working at Ecotron facility of Hasselt University, which belongs to the ANAI. And what is an Ecotron? It is a set of advanced state-of-the-art climate chambers where we can put very large samples of an ecosystem. So in our Ecotrons, we have lysimeters of 1.5 meter depth and 2 meters in diameter, with above ground part being more than 5 meters high and also very large. So you could put considerable sample of an ecosystem in such climate chamber, and then you can impose climate of future or of a past with very high level of details. We don't only control the temperature. It's easy to say future climate, there will be high temperature. Let's increase temperatures by 2 degrees. We do this, but not only this. We play entire climatic scenarios. So we have scenarios prescribing the weather of every hour for a future climate, and we are playing these scenarios in our Ecotron, controlling temperature, precipitation, wind speed, atmospheric pressure, also parameters of a soil, also temperature, water availability, water tension. All these parameters are controlled and also monitored with very high level of resolution. Some things are monitored every minute, as you see, some things maybe every 10 minutes, every half an hour. All this together allows us to have a system where we can follow ecosystem functioning with a very high level of detail, which of course creates very nice system to study phenology and phenological mismatch. In relation to PHENET, we are working with two case studies, both run in the ecotron, so it's two independent experiments. On each of the experiment, PhD student is working. The first experiment is about functioning of pear orchards in future climate, where we have very simple experiment. We subject pear trees, adult pear trees to climate of 2044 in comparison to the current climate. So our control is current climate. Seeking answer to the question how the pears will be functioning in future, pear trees will be functioning in future climate. Will pears still be abundant? Can we still have normal harvests? Will they be of good quality? And what will be the processes in the how climate change will affect processes in soils of pear orchards. So here we are using six items, units of the ecotron. In 3 units, we have 6 trees in a climate of 2044, and in other 3 units, we have 6 trees of in climate current 2018. And a climate of 2044, the scenario is predicted based on RCP predictions of increase in atmospheric carbon. We use the most severe prediction of RCP 8.5. And based on the predictions of the atmospheric carbon level for 2044, we have a climatic scenario predicting the entire climate regime of 2044 played hour by hour, day by day. And we see indeed that above ground, we observe very obvious phenology changes. You see above in the upper picture, photograph made in 25 of April in a climatic scenario of 2040. And you see flowers on pear trees. And on exactly the same day in climatic scenario of 2018, so actually it is the same day, the years refer to the climatic scenarios here, pear trees don't have any flowers yet. And we see it in other measurements like discoloring of the leaves, fall of the leaves, etc. Oops. Yeah. Another case study which we run is about natural ecosystem. We are investigating impacts of climate change on below ground processes in heathland, and why it is still relevant for PHENET project, because in a country like Belgium, we are living actually in a very tight mixture of natural ecosystems and agricultural ecosystems. So our heathlands are actually bordering ecosystems like pear orchards. And here we are specifically focusing on below ground carbon allocations and below ground processes, but we address climate change in a little bit different way. Here we are asking question how severe should be climate change to lead to cause strong changes in ecosystem functioning of heathland that would be so strong that heathland actually would turn into other type of ecosystem. So from shrubland type of landscape located on nutrient poor acidic soil, it would change maybe into grassland. So we are asking how strong should be climate change for this to happen? So to answer this question, we are using also six climatic chambers in our ecotron, but then in every climatic chamber we have its own climate, spanning from the past climate when there still will be no climate change manifestations of 1950 to current climate and to future climate. And every climatic scenario is also used on RCP predictions of scenario 8.5. Well, so in this case, we were taking this scenario, RCP 8.5, examining which levels of carbon dioxide increase is expected at given year, which we are interested in, and developing climate scenarios for that year, and then playing it in the climate chamber of the ecotron. So these are the two study cases which we are working with, and now I'm going to speak about research questions which we have and discuss how do we process the data in context of PHENET and what kind of services we are developing and what kind of services we would like also our colleagues at PHENET to develop to help this type of research to be conducted in NA facilities. Well, first about our part of work, our own service development. I have mentioned that we are interested in comparing different climate scenarios. So we work with 6 different climates from past to future. It sounds very easy. We compare different climates, but very stupid and naive question. What do we actually compare? If we want to have a quantitative predictor describing severity of a climate change, what can we use as such a predictor? We cannot use the year which we are for which we developed this climatic scenario. So comparing number of 1950 to value number of 2030 doesn't make any sense, right? What we could theoretically do is to use as a quantitative predictor the value of carbon dioxide concentration, which is expected in each of these years. Yes, that's possible, but climate change is much more than only change in carbon dioxide. We could use as a predictor average increase in temperature. But again, climate change is much more than climate warming. It's also change in precipitation regime. It is changing number of dry days, freezing days. So the solution which we have developed is to work with climate indices. I give here an example, but actually we work with 20 climate indexes, such as for every climatic scenario, we calculate number of dry days, number of freezing days, coldest winter temperature, warmest summer temperature, and other things which describe the climate. And then we subject this entire set of climate indices to LDA analysis, linear discriminant analysis, and that allows to locate every climate in a multidimensional space of climate indexes and allow to derive for every climate only the 2 values which is positioned on LDA axis 1 and 2 as a predictor of severity of climate change. And that's our approach which we systematically apply to all our analysis. So our predictors when we need a quantitative predictor of severity of climate change, is this positioning of the climate, of the climatic scenario in multidimensional space of climatic indexes. Another type of service which we are developing based on this work is impact of climate change on ecosystem functioning. So here you see example of impacts of climate change on abiotic soil conditions. So an ecotron allows constant measuring of temperature, of soil temperature, soil moisture content. So this kind of data we also able to provide to our users. Well, now a little bit about our results. What we see is that in future climate, ecosystems like pear orchards actually accumulate carbon. So in this way, they act as mitigators of climate change. We have very simple explanation in case of orchards, but not in case of heathland. I'm going to talk about it with the next slides. In orchards, it's very easy. Indeed, ecosystem accumulates more carbon because there is more photosynthesis. And why there is more photosynthesis? Because it is warmer, we have more higher number of growth days. Plants don't only start to flower earlier. The picture which I have shown you, they also the bud burst starts earlier. The leaf fall is later. So altogether, plants have photosynthetic are able to photosynthesize longer, and therefore they are able to assimilate more carbon. In case of heathland, we see actually exactly the same pattern. So in future climate, heathland assimilates more carbon, but interestingly, we don't see any increase of above ground biomass. Even stronger, we see decrease of above ground biomass. So carbon is accumulated in the entire ecosystem, but not in a plant biomass, not in a visible part of plant biomass. We also don't see any increase in carbon concentration in leaves and branches of plants, neither in roots. So leaves still have the same amount of carbon as they would have normally, as they would have in the current climate. But what we do see is that a lot of carbon is allocated below ground into the roots and into the microorganisms. We see similar patterns in pear orchards, and there we are asking a question into which microorganisms actually this carbon is going, and especially we are interested in impact of climate change on mycorrhizae. That's a fungi, well, we have discussed it with previous speakers. That's a fungi living in symbiotic relationship or with roots. So we are investigating how climatic change affects community of mycorrhizal fungi in roots and beyond the roots. And because we are interested ultimately in phenology, we are interested how this carbon allocation between different seasons in autumn and summer, in a very late autumn, how these patterns are affected in different climates. Well, we are also interested in impact of climate change on other soil microorganisms, and we see that there is indeed quite strong effect. In future climate, we will have different microbial communities than in the current climate. And if we look at individual microbial groups, that's what we are also interested in. We can detect microbial groups which are affected stronger by climate change than the other ones. What we see, for instance, that bacteria are affected stronger than fungi, and mycorrhizal fungi in heathland are, for instance, not affected at all in terms of community composition. You still have the same fungal species growing there. About the biomass, we don't know yet, and that's where the tools that Pavel has been discussed would be extremely interesting, for instance. Well, the next research question which we are going to ask in relationship to mycorrhiza is whether in future climate mycorrhizal fungi will remain mycorrhizal, especially so mycorrhizal fungi which are present in the heathland ecosystem are able to also conduct saprotrophic lifestyle. That means that if they don't need their host plants, they could disconnect from the host plants and start to acquire carbon from organic matter, decomposing an organic matter. In future climate, we see that mycorrhizal fungi receive more carbon from their host plants. That's logical. Plants are assimilating more carbon, and they are also sharing it with their symbiotic partners. If you are mycorrhizal fungi and you're already getting all your carbon, normally speaking, from a plant, and now you're getting even more carbon, what you would do? Would you stay with such a nice host which is feeding you with the carbon? That's personally I would do if I would be a mycorrhizal fungi, because I'm lazy, and if I get the resource, I would like to use it. However, there is alternative theories from my colleagues who like to work. They say if mycorrhizal fungi get more carbon, they get more resources. That means that they have more capability for saprotrophic lifestyle, and they will start living saprotrophically. Why we are so interested in it? Of course, from fundamental biological perspective, but also from very applied perspective of decomposition of organic matter. If mycorrhizal fungi in future climate, by receiving more carbon from plants, start to have saprotrophic life mode, that means that they will intensify decomposition of soil organic matter, releasing more carbon into atmosphere, and therewith further accelerating climate change. If mycorrhizal fungi stay mycorrhizal, they simply receive more carbon from the plants, but all this carbon is staying in the soil. This entire pathway reduces carbon dioxide in the atmosphere and therewith mitigates climate change. So these two potential pathways of development of mycorrhizal fungi lead to two absolutely opposite consequences, scenarios of ecosystem response to climate change and of a feedback. Therefore, it's extremely interesting question. Well, given all this analysis, how we would like to further benefit from PHENET, we are able to examine responses of microbial communities in terms of acquiring OTU tables, and I have seen you some pictures which probably was very difficult to interpret. You see communities are different, but in what aspects they are different, it's very difficult to say. So we would like to get better visualization tools which are of a type which I show at the right side of the slides that our colleagues have been developing, showing allowing visualization of treatments conducted in the ecotron on microbial communities. Well, another aspect what we are interested in is assessing enzymatic activities in soil. Ilya has been talking about a little bit. So idea here is that we have specific fluorescence labeled substrates that are added to soil slurry, and then we use high throughput measurements of fluorescence release in in 96 well plates to measure activity of individual enzymes. And our results so far showing that despite obvious impact of climate change on microbial community composition, we don't see change in enzymes produced by these microbial communities. However, now we are limited to one moment measurements of enzyme activities. We are taking the soil, go to our machine and examine the enzyme activities. What would be much more interesting is to have continuous measurements of enzyme activities, and therefore we are looking for possibility to use long-term monitoring devices there. And I know those are being developed. We are looking at this currently, whether we could use kind of sensors measuring enzyme activities in the real time. Well, the last part is the project, sub-project, which my PhD student is only starting now. We are looking at climate change effect on plant root phenology, and therefore we are indeed using the rhizotrons, which Ilya has mentioned. We install them in our ecotron at different depths that allows us to monitor development of roots. And further, we would be indeed extremely interested to also monitor development of mycorrhizal fungal communities, or at least mycorrhizal fungal biomass. The rhizotron cameras which we are using now are not able to see the mycorrhizal fungi, but perhaps with a group of BOCO we could collaborate and get maybe better device or better camera here. Well, and the last slide, the last wish for the PHENET input. We are taking these multiple measurements individually. Each of them is already pretty complex and involves visualization, interpretation, and understanding the data. But altogether, it would be very nice to have a joint understanding of dynamics of the changes in different parts of below ground communities. So roots, microbial communities, abiotic conditions, how to visualize their changes and their dynamics jointly so that we see the relationship between activity of different organisms and different drivers. Well, this was the end of my presentation. Thank you for your attention. Now I'm ready to answer questions.